So what happens in automated Dye-Terminator DNA sequencing? இது நம்ம என்ன பண்ண போறோம்? Automation பண்ண போறோம். Okay. We humans are very lazy. And our laziness is what gives rise to so many inventions. Like that, when we found out that manual is going to be very tedious process, we found out a way to automate it. So in that, what we will do is, the dideoxy DNA sequencing was time consuming, radioactive, as well as the throughput means the outcome. Okay, throughput was low, typically 300 base pairs per run. Or run we can only do for 300 base pairs. Okay, so automated DNA sequencing employs the same general procedure, but it uses ddNTPs labeled with fluorescent dyes. So instead of radioactive things are dangerous for our health also. Okay, so instead of radioactively labeling these ddNTPs, we will label it with fluorescent dyes. Fluorescent means in a light emitting. Radiation is different, fluorescence is different. Okay, so in this we'll be using fluorescent dyes. So combine the 4 dyes in one reaction tube and electrophorese in one lane on a polyacrylamide gel or capillary containing polyacrylamide. So another difference is, first difference is in automated we are using fluorescent dyes. Okay, one second. We are going to use the fluorescent dyes instead of radioactive dyes. Second difference is, we'll combine all the 4 dyes into one reaction. Okay, in another one we had 4 reaction tubes. Idhila particularly we added DDA, here DDC, DDG and DDD. Now what we'll do is, we'll label all these DDATPs with different fluorescent dyes. So you know if this color is coming, it will be A. If some blue color is coming, it will be C. If some green color is coming, it will be G. Like that, we'll differently label these DDNTPs with different fluorescent dyes, put it inside a single tube. Okay, one reaction tube. And so we'll need only one gel in the electrophoresis also. And we'll be running it in something called a capillary. Capillary is a very thin tube. Okay, and inside that capillary, the gel is filled. And through that gel, on top, we will put our reaction mixture, and it will be coming out. According to size, it will be separated. And once this comes out, right, these fragments containing fluorescently labeled DDNTPs, where and all this has been terminated, those small pieces, according to size, will come down. And once they're coming down, at the point where it is coming out, it will be, some rays will be passed through it. Okay, some rays will be passed through it, and we will be able to understand which color it is. So correspondingly, which nucleotide it is. I'll show it to you with a diagram later. So UV laser rays. So we are using UV laser rays. Okay, so UV laser detects dyes. So it's basically a dye, that is, color is nothing but dye, right? UV laser detects dyes and reads the sequence. Sequence data is displayed as colored peaks on chromatograms that correspond to the position of each nucleotide in the sequence. Throughput is high. Throughput means, again, I showed the outcome. In Sanger's manual method, we could only do 300 base pairs in one single gel run. But here in this capillary tubes, you can do up to 1,200 base pairs per reaction. And 96 such reactions can be done in every 3 hours with capillary sequences. It's like made our life much more simpler, or it has made our life much more faster. So most automated DNA sequences can load robotically and operate around the clock for weeks with minimal labor. So you don't even have to load the samples. Everything is done by robots. So this sequencer will be like going on for weeks and weeks and weeks. So after one sample, the other sample, and it will go on take automatically. Okay. So this is a small diagram that is shown. DNA sequencing with fluorescent dyes attached to chain terminator dideoxyribonucleotide. So you can see they are labeling the particular DDATP or CTP and GTP with particular fluorescent dyes called fluorochromes. Okay. Chrome is nothing but color. Fluorescent, fluoro is for representing the fluorescence. Okay. You're going to do the same thing, everything together, DNA polymerase, DNTPs, primers, as well as the DDNTP, small amount, will add in a single test tube with your DNA template. And your chain will extend, and wherever your particular DDCTP or DDGTP is coming, what will happen? It will terminate the chain over here and you will get different chains. Now you might have a doubt, Appo, why did we have separate tubes for manual ones? uh, can you think of why അതിലേം இந்த மாதிரி the same things we were using, then why is 4 separate tubes, and why can't we add all the DD NTPs together? Why were we adding in one tube DD ATP, one tube CTP and GTP? The basic difference is in that the same radioactive substance was put up in these ATP, CTP and GTP and ATP. புரியுதா? அப்போ என்ன ஆகும்? சேம் இருந்தா all will look the same to you. You will not be able to separate it. That's why we needed 4 different tubes, and one we needed to put ATP, separately 4 nucleotides. But here we are labeling it with different colors, different fluorescent colors. So we can put it all together. So whichever color is showing up, that is our NTP. Okay, that is our nucleotide. So this is a diagram that is showing. So you will load your sample over here. This is a capillary tube. Okay, capillary tube means a very thin tube. Inside this, your polyacrylamide gel will be there, and as it passes through it, at according to the size as it goes down, there will be a detector over here which will pass a this laser will be passed from here. A detector will catch what is the peak that we are getting, and you will get something like this. So these are of different colors. I hope you can see the different colors. If blue is coming, whatever nucleotide is colored as blue will be the one. For example, they are saying it is C, C, T, like that. Okay. According to different colors, you will have different peaks. And then it will generate the sequence like this. So it is all automated. There is no tension. You don't have to even sit and see the colors. Everything will be done by the computer. So the resulting colored DNA fragments are separated by size in a single electrophoretic gel lane. So here only one gel lane is used. All fragments of given length migrate in a single peak. Okay. The color associated with each peak is detected using a laser beam. The DNA sequence is read by determining the order of the colors in the peaks as they pass through the detector. As they pass through this point, they will all flow down at one point. They'll start flowing from here. So one by one, as they pass, they are being detected, and you get a chromogram over here. From that, you can understand the sequence. This is again another diagram that is showing the same thing. Okay. This will be connected to the computer. You will get the data. This is how that electropherogram looks like. Okay. Before I go into the pros and cons, let me just ask you guys. This is what is Sanger's dideoxy chain terminator method, in which you have manual method and automated method. I hope both the methods are clear, because if you have understood manual, it is very easy to understand automated, because only a few changes we are making. That's why we could complete it so fast. Yeah. We'll just go through the pros and cons. Pros means advantage, cons means disadvantage. Okay. So pros and not as toxic and less radioactivity than maximum Gilbert. Maximum Gilbert is the next method that we are going to learn, chemical degradation method. Okay. அப்ப அதோட இது பார்க்கும்போது கொஞ்சம் கம்மி டாக்ஸிக்கா இருக்கும் and கொஞ்சம் கம்மி radioactive இருக்கும். Okay. Then it is easier to automate. Idhili automation is possible, right? நம்ம படிச்சோல எப்ப automation is possible. But maximum Gilbert method la automation is not possible. Okay. And it is almost highly accurate, and it is easier to get started. Now, what are the disadvantages? Poor quality in the first 15 to 40 bases of sequences. This is due to primer binding. First konjam primer binding ku nammal space vidanolla and the sequence namukku theriyamaattom. Wherever the primer has bound, அதோட opposite இருக்கிறது synthesis நடக்காதுல்ல. So you will not be able to find out that sequence. So if you want to find a whole genome, in between wherever primer has joined that sequence, you will not have a clarity. Okay. Then it is time consuming, especially due to requirement for electrophoretic separation of fragments. Electrophoresis takes some time. Okay. Then it is expensive due to relatively large volume of chemicals that are used. So many chemicals are used, it is expensive. Okay, so that's it, I guess. So this is the lab setup. Okay, you can see a sequencer over here, which is connected to your computer. I just put this picture so that you get some idea how it looks like actually. These are different sequencing machines that are used in